The Greater Blue-Ringed Octopus, otherwise known as Haploclana lunulata. This deadly sea creature is known to be among the most deadly of all cephalopods. Known to carry one of the most deadly toxins known to man, this octopus is capable of killing up to 26 fully grown men all by itself. This animal belongs to the Animalia kingdom of the Eukarya domain, phylum Mollusca, the class Cephalopoda, the order Octopoda, the family Octopodidae, the genus Haploclana, and it's the species Haploclana lunulata. This octopus species lives mostly in coral reefs and rock pools at a depth of about 0 to 20 meters. The greater blue-ringed octopus's diet consists mainly of crustaceans, mostly crabs, and it hunts these by using a very special type of poison unique to its genus. The reason that H. lunulata, the greater blue-ringed octopus, is called greater among the 4 confirmed species of the Haploclana genus is not because it is larger than any of the others, but because its blue rings are the largest among the genus, which is its main phenotypic feature. My name is Shahan Pereira, and today I'll be talking to you all about this octopus species. Specifically, I'll be talking about features it has evolved that other octopus species don't. I'll be doing this by analyzing its biogeography, its homologous and analogous structures, its vestigial structures, and any adaptations and variations it has that differ from other octopus species. So let's get right into it. Alright, let's start by looking at a worldwide map so that we can get a general idea of where the greater blue-ringed octopus species can be found. As we can see on the map, they're found in a relatively confined area of the world that consists mostly of islands, especially small ones. Now let's look at a zoomed in version of the area where they can be found. As we can see clearly now that the greater blue-ringed octopus species lives near small islands at a shallow depth of around 0 to 20 meters where rock pools and coral reefs are very common. In terms of the geographical distributions, these areas, as indicated by the red dots, are northern and southern Australia, as far north as Japan, which includes the Papua New Guinea and Solomon Islands, as well as Indonesia, and they extend as far west as Sri Lanka. Alright, let's move on to homologous and analogous structures now. Alright, let's start with homologous structures first. The first one we'll be looking at is that all octopus species, including the greater blue-ringed octopus, have 3 hearts. This is to cope with the fact that their blood is a poor carrier of oxygen. The reason for this we'll get into later. 2 of these hearts circulate blood to the gills, whereas the other one circulates it throughout the body. This also maintains a high blood pressure, which helps to cope with the low oxygen levels in their blood. Alright, let's look at the blood itself now. Octopus species all have blue blood instead of red blood like us humans. This is due to the fact that their oxygen binding agent does not contain iron, which would make it red. Instead, it contains copper, which makes it blue. Their oxygen binding agent is known as hemocyanin, whereas ours is known as hemoglobin. Alright, the next 2 homologous structures are closely related to each other, so we'll talk about them together. The first are a special type of cell known as chromatophores. Chromatophores are cells that contain 3 sacs of color each. There are tens of thousands of these cells cover an octopus's skin, and muscle contractions on top of and around these cells help change the way that colors are expressed, allowing an octopus to hide themselves in their surroundings. Each chromatophore is controlled individually by the nervous system, allowing for very high accuracy mimicry and instantaneous response to the environment. The second homologous structure related to this is iridophores, another type of cell. These cells are reflective in nature. They help to camouflage the octopus by mirroring the surrounding environment's colors, which allows the octopus to mimic the environment to an even higher degree of accuracy in order to avoid predators. Alright, let's look at analogous structures of the greater blue-ringed octopus in comparison to the rough-skinned newt. These 2 organisms are very unrelated to each other, as according to their taxonomic classifications, their closest relationship lies in the fact that they both belong to the kingdom Animalia, which means they split off a very long time ago. Despite this fact, both of these organisms use a very similar self-defense mechanism, which is to use the poison or toxin known as TTX, tetrodotoxin. This is a self-defense mechanism that both organisms use. They produce this toxin in various glands under their skin or inside their bodies. These glands house colonies of TTX producing bacteria. This is a symbiotic relationship that is once again analogous. Many other organisms show this analogous relationship as well, such as tapeworms and a pufferfish, which uses the same type of poison. Now let's look at a vestigial structure of the greater blue-ringed octopus. The vestigial structure we'll be looking at is the ink sac. This may come as a surprise, seeing as most octopus species use the ink sac as their primary self-defense mechanism and anti-predator mechanism. The greater blue-ringed octopus has partially lost their defensive ability to ink, as their ink sac has atrophied as they grew up. The ink sac in H. lunulata is vestigial, therefore. And although paralarvae of the species can produce minute amounts of ink infrequently, it is no longer a dependable anti-predator mechanism, and they therefore rely on TTX as their primary anti-predator mechanism. Now let's look at some adaptations and variations of the greater blue-ringed octopus in relationship to other octopus species. The main adaptations and variations of the greater blue-ringed octopus, H. lunulata, lie in the fact of its use of two separate types of toxins, TTX, tetrodotoxin, and haplotoxin. TTX is an extremely potent toxin with no antitoxin. It is produced in the two posterior salivary glands by symbiotic bacteria. This toxin is only released as a self-defense mechanism and is extremely harmful to humans. It is said that 1 milligram of this toxin is enough to kill a human. This is both a physiological and behavioral adaptation. The second primary adaptation of the greater blue-ringed octopus lies in its second toxin, haplotoxin, named for the fact that it is only produced by species of the genus Haploclina. This toxin is used for hunting and is released by the octopus into surrounding water in the vicinity of crabs and other crustaceans, causing paralysis. This way, the octopus can dismember the crab before consuming it. This allows the octopus to feed on relatively large prey without significant risk of self-harm. Well, that's all. Now, we can see by analyzing the biogeography, the homologous and analogous structures, the vestigial structures, and the various adaptations and variations of H. lunulata, we were able to see and glimpse into the evolutionary timeline of this organism and see how it's adapted over time to various environmental and other factors such as predators by developing new anti-predator mechanisms and for new structures for coping with various environments. Thank you for watching and have a good day.